scientists have no idea where it came from. What they do believe is that if you have that, you may have been evolved from a different kind of species on this planet.
Think of human blood like a locked door with 4 keys, A, B, A, B, and O. For a century, we thought we had the only keys that worked. Well, AI just picked the lock. It found a 5th key, a secret blood type that's been hiding in our DNA for thousands of years. It's being called Elytra, and it's turning hematology upside down. Some people with this blood seem almost superhuman, immune to modern plagues. But what most people don't realize is the devastating secret that comes with it. When the AI outsmarted science. The whole thing started in a chilled, humming server room in Paris. The date was July 2025. A team of French hematologists and data scientists were running a new experimental project. They were trying to solve the problem of rare blood. So they fed 2.5 million anonymized medical records into an artificial intelligence. This wasn't just any AI, it was a cutting-edge deep learning system, a convolutional neural network. Basically, a digital brain modeled on our own, designed to find patterns that humans would miss. They cross-referenced all this data with AlphaFold 3, another AI that can predict the exact 3-dimensional shape of almost any protein in the human body. The plan was simple. They'd let the machine sift through terabytes of data. We're talking genetic sequences, blood test reports, patient histories, everything. They were hunting for new, ultra-rare donors. You see, what most people don't realize is that the whole ABO system is just the front door. It's a massive oversimplification. That system, found by Carl Landsteiner way back in 1901, is all about antigens. Think of them like little flags on the surface of your red blood cells. Type A has the A flag, Type B has the B flag, Type AB has both, and Type O has neither. A few decades later, in 1940, the same scientists found the rhesus factor, that's the plus or minus, the positive or negative next to your blood type. So we all settled on 8 main types. Easy. But here's the catch. That's not the whole story, not even close. Science now knows there are over 360 different antigens organized into 45 different blood group systems, and some of them are incredibly strange. Everyone's obsessed with the Bombay phenotype, found in 1952. These people are genetically AB or AB, but their tests always come back as O. Why? Because they're missing the H antigen, the hook that the A and B flags need to attach to. Without the hook, the flags just float away. The crazy part is, these people, about 1 in 10,000 in India, can only get blood from other Bombay types. Give them regular type O and their body attacks it. And then there's the real rare stuff. Golden blood. Its real name is Rh null. These people are missing all 61 antigens in the entire Rh system. Not just the one minus antigen, but all of them. For decades, doctors thought this was impossible. They thought a person couldn't survive without those proteins. But they do. Fewer than 50 people on Earth are known to have this golden blood. It is the ultimate universal donor. But the people who have it can only receive blood from another Renault person. They are walking on a biological tightrope every day of their lives. So, the French team knew our understanding of blood was shaky. They were just hoping to find a few more variations. For weeks, the AI just sorted data. It categorized, it learned, and then it flagged an anomaly. It wasn't a mistake, it was a pattern. The AI spat out a list of 47 names, 47 people from all over the planet. These 47 people did not fit any known category. They weren't A, B, A, B, or O. They weren't Bombay. They weren't Renault. They were something else. The machine had found a spectral signature, a molecular fingerprint so rare and so weird that every time it had popped up in a lab, a human had dismissed it. Testing error, they'd write, contaminated sample. But the AI, with its massive non-human brain, saw the pattern across millions of files. It knew this wasn't an error. It was a group. The lead scientists, staring at the results, were stunned. They thought the AI was broken. They ran the data again. The same 47 names popped up. The AI had found what looked like a 5th blood type. They gave it a provisional name, a code name for the project, Elytra. It's a term for the hardened protective wing cases of a beetle A shield. They chose that name for a reason, because this new blood type seemed to be shielding its owners from some of the worst diseases on Earth. But what they didn't know yet was that this shield was a double-edged sword. The AI had given them a list of 47 names, but to understand the discovery, they had to start with one single impossible blood sample frozen for 2 years in the Caribbean. Marie's secret. The first name on the AI's list, the index case, was codenamed Guada 1. Her real name was protected, but they called her Marie. She was a 42-year-old nurse living in Guadeloupe, a French Caribbean island. Marie had given blood during a routine drive in 2023, almost 2 years before the AI project even began, and her sample had caused total chaos in the local lab. A young technician had put her vial into the centrifuge, the machine that spins blood to separate it into layers, but when it came out, something was wrong. He called his supervisor. The serum, the clear yellowish liquid part of the blood, had clotted, but not in a normal way. The supervisor later described it in a panicked report. It clotted like alien ichor. They ran the standard tests. No A antigens, no B antigens. Okay, so she must be type O. Easy. They tested her for the H antigen just in case she was a Bombay type, but the test came back positive. She had the H antigen. This made no sense. It was a biological contradiction. How can you have the hook, the H antigen, but not have any flags, A or be a normal O type? It was like having a foundation for a house, but no house and no empty lot either. It was just wrong. Flustered, the lab techs did the only thing they could. They labeled the sample untypable. They wrote testing anomaly, possible contamination on the file, and they put the vial into a deep freeze at minus 80 degrees Celsius or minus 112 Fahrenheit. And there it sat, a tiny frozen puzzle box, until the AI in Paris 2 years later dug up its data file. The French team contacted the hospital in Guadeloupe immediately. This wasn't a request. It was an urgent demand. They needed a fresh sample. Now, Marie, the nurse, was baffled when she got the call. She was a healthy, active mother of 2. She was on her feet all day at the hospital. She felt fine. She agreed to come in. The scientists, now on high alert, flew a special team to the island. They didn't just take one vial, they took 20. They ran her blood through every test imaginable. Flow cytometry, mass spectrometry, genetic sequencing, cryo-electron microscopy. The results started to paint an unbelievable picture. First, her genetics. The AI had already flagged this. Her mitochondrial DNA, the code passed down only from the mother, was haplogroup L3E, a common lineage tracing back to the Bantu expansions in Africa about 3,000 years ago. Standard stuff. But her paternal Y chromosome was where things got wild. It was a lineage called E1B1A, but it was littered with ghost DNA. And get this, the AI identified the ghost DNA as a 99% match for Melanesian Denisovans. Let's break that down. Denisovans were an ancient cousin of humans like Neanderthals who lived in Asia tens of thousands of years ago. We thought they were just a footnote in history, known only from a few bone fragments and a tooth found in a Siberian cave. But their DNA, this ghost DNA, is still found in tiny amounts, about 3 to 5%, in people from Melanesia and places like Papua New Guinea and the Solomon Islands. What was it doing in a nurse from Guadeloupe whose family had been on that island for generations? This wasn't a single mutation like the one for sickle cell. The AI proposed something crazier. It was an epigenetic symphony. Basically, Marie hadn't just inherited this blood type. She had inherited a complex set of on-off switches for her genes, and something in her environment, the AI speculated it could be a specific rare fungus in the local soil or even long-term exposure to a mineral in the water, had flipped all those switches in a perfect 1 in a billion sequence. It was like she had unlocked an ancient genetic program, a program that came from ancestors who lived 50,000 years ago. The blood itself was even weirder. The scientists put it under a cryo-EM microscope, a machine that freezes samples and blasts them with electrons to see their shape. On the surface of her red blood cells, they saw it. A brand new, never-before-seen chimeric glycoprotein. A glycoprotein is just a protein with a sugar chain attached. But this one, which they dubbed ABCDE1, was a monster. It was built from pieces of the normal ABO scaffolds, but it was capped with a bizarre helical chain of sialic acid. It formed a shape they called the Elytra coil, and that coil did something stunning. It twisted the antigens, folding them over and hiding them from view. It was a molecular invisibility cloak. It was hiding the H locus. That's why her first test was so weird. She had the H antigen, but the machine couldn't see it. It was hidden under the cloak. This genetic ghost story wasn't just in her past, it was rewriting her present. The Elytra coil was a shield, and that shield was protecting her, but it was also slowly failing her. The price of survival. The discovery of the Elytra coil set off a firestorm of new experiments. The scientists in Paris were looking at something that shouldn't exist. This invisibility cloak was a game changer, and here's the deal. It didn't just hide the blood type from lab tests, it seemed to hide it from diseases. The team, now in a state of barely controlled panic, started running in vitro tests. That means tests in a lab dish. They took Marie's plasma, the liquid part of her blood, and mixed it with pseudoviruses. These are harmless, disabled copies of some of the most dangerous viruses on Earth. The results were mind-blowing. They started with SARS-CoV-2, the virus that caused the global shutdown. Normal plasma can neutralize some of it, but Marie's Elytra plasma was a super weapon. The Elytra coil, with its hyperglycosylated decoys, those complex sugar chains, acted like a biological fly trap. It bound to the virus's spike proteins and neutralized them 87% more efficiently than even the best vaccinated plasma. It was like the virus couldn't even see her cells. They got bolder. They tried Zika, another virus that plagues the tropics. Elytra plasma cut the virus's ability to invade human cells in half. They tried a strain of H5N1, the bird flu. The plasma stopped it cold. This was stunning, but it was all in a lab. What about in a real person? The AI's list of 47 donors gave them the answer. The team started cross-referencing the patient histories, a grim task of digging through other people's lives. They found 2 cases that made their blood run cold. One was a fisherman from Martinique. The other was a village elder from the Solomon Islands. Both men were on the Elytra list, and both men's medical files showed decades of high-risk exposure to HIV, yet both men had 0 HIV titers. They had never gotten sick. They were completely, mysteriously immune. It was as if the virus, one of the most clever and deadly on the planet, hit their T cells and just bounced off. They had shadow immunity. This was the miracle. This was the shield the name Elytra promised. This ancient blood type, this ghost DNA, was potentially a key to immunity from the worst plagues of the modern world. But not all things are what they seem. The scientists started digging deeper into the 47 files, and they found the other side of the paradox, the curse. 2 of the carriers, one in Papua New Guinea and one from the Basque region of Spain, had passed away. Their cause of passing was listed as complications from immune failure. The team looked closer at Marie's blood work. She felt healthy. She was a nurse. She was active, but her numbers told a different story. She had chronic lymphopenia. That's a fancy way of saying her T cells, the soldiers of the immune system, were disappearing. And get this, the AI calculated the rate. Her T cells were eroding at a rate of 15% per year. This was the horror of Elytra. The invisibility cloak was too good it hid the red blood cells from viruses, but it also seemed to confuse the body's own immune system. The T cells, designed to hunt and destroy invaders, couldn't find anything. The cloak was so effective it made her body look sterile. The T cells were essentially dying of boredom. They were wearing themselves out, aging and fading away at a terrifying speed. It was like accelerated aging, but only for the immune system. And then there was the transfusion problem. What if Marie or any Elytra carrier got in a car accident? What could they give her? They ran the simulation. The Elytra blood in silico in the computer looked like a perfect universal donor. It had no A or B antigens, so it should work for everyone. But when they tested it in vitro against standard plasma, the reaction was catastrophic. Anaphylactic cascades. The test plasma lit up with immunoglobulin E or IgE. Those are the antibodies that cause deadly allergic reactions. The IgE spikes were 300% higher than even the violent reaction from a Bombay-type mismatch. Giving Marie a standard blood transfusion wouldn't save her. It would end her life in minutes, and giving her blood to anyone else would do the same. They were a fifth blood type. And they were, in every practical sense, totally alone. This paradox wasn't just a medical curiosity, it was a global mystery, and it was about to get a whole lot weirder. The AI's origin model was ready, and it pointed to something impossible. The Atlantean survivors. The 47 donors were a puzzle that defied geography. The AI had plotted them on a map. A nurse in Guadeloupe, a fisherman in Martinique, a village elder in the Solomon Islands, an isolated community of Ainu in northern Japan, several members of an Ashkenazi Jewish family in New York, a Basque shepherd in the Pyrenees Mountains, a member of the Yanomami tribe deep in the Amazon rainforest. How? These populations had zero contact with each other, no shared migration routes, no shared family trees. What most people don't realize is that our DNA is a roadmap of our ancestors, and these maps were all pointing in different directions. But the AI saw the one connection, the ghost DNA. Every single one of the 47 carriers had a strange, unique mix of ancient human DNA. Neanderthal, Denisovan, and other ghost populations scientists haven't even named yet. Marie's Denisovan link wasn't a fluke, it was the key. The AI ran a new model. It crunched 10 to the 6th power epitopes, that's a lot of data points, to find the origin. The answer it spat out was stunning. The Elytra blood type wasn't new, it was ancient. The AI pegged its emergence at roughly 5,000 years before Common Era. That's over 7,000 years ago during the Neolithic period. This was when humans were first settling down, starting to farm, and building the first large towns. The theory? This epigenetic switch first flipped on in one of these new dense populations, and from there it spread. It wasn't a dominant trait, so it stayed hidden, passed down silently. It spread along the ancient Silk Road, which explains the carriers in the Basque region and in the Ainu of Japan. It spread through the Bantu expansions in Africa, which explains Marie's L3E haplogroup. It spread across the Bering Strait into the Americas, leading to the Yanomami carrier. And it spread through the complex Atlantic slave trade, which is how a gene signature from Melanesia likely ended up in a woman from Guadeloupe. The world was more connected, more anciently than we ever knew. But the most incredible wow factor came from a tiny cluster of islands in the Indian Ocean, the Andaman Islands. The French team, guided by the AI, reached out to researchers working there. The Andamanese people are one of the most isolated populations on Earth. They have lived on those islands for tens of thousands of years. The researchers tested the local diver population. The results were staggering. In the general world population, Elytra was 1 in millions. In this isolated tribe of divers, it was 1 in 500. The tribe, it turned out, already knew. They had folklore about it. They called it spirit blood. Their elders told stories of certain families whose blood danced with ghosts. They said this blood gave them special gifts. They claimed it gave them sea vision, an uncanny ability to see clearly in the deep, murky water while diving for food. This, scientists now guess, could be a neurological side effect of the Elytra glycoproteins, or maybe something else. Some fringe biologists are even linking it to the old aquatic ape hypothesis, the idea that humans spent part of their evolution living in water and that Elytra is a reactivation of those ancient aquatic genes. But the folklore also had a warning. The spirit blood came with a curse. The elders said those with the spirit blood were often cursed with fertility. They had trouble having children. This was a perfect, heartbreaking match for the data in Paris. The chronic lymphopenia, the T-cell erosion, the accelerated aging of the immune system was likely causing infertility. The tribe had known for centuries what science was just discovering. This blood was a gift and it was a curse. But this is where the story goes from science to something else entirely. because the AI's origin model, it had an alternative. The Denisovan match wasn't 100%, it was 99%. The AI flagged the remaining 1% as unidentifiable, and that 1% has opened the floodgates to theories that are making scientists' hair stand on end. Wild theory number 1, the Anunnaki experiment. This theory is a favorite of ancient mystery buffs. It says that ghost DNA isn't from Denisovans at all, it's non-human. The theory points to ancient Sumerian texts, the oldest in the world. These texts describe gods called the Anunnaki who came from the sky. The stories say the Anunnaki created modern humans by mixing their own essence with that of early hominids. Why? To be slaves, to mine gold for them. So here's the deal. What if the Elytra blood type is the evidence? What if it's the royal blood of the Anunnaki gods, a genetic marker they left behind? Or even darker, what if it was a failed experiment? A prototype human, version 1.0, with built-in immunity but a fatal flaw, the T-cell erosion that made them useless as workers. What if the 47 carriers are just the last echoes of a divine experiment gone wrong? Wild theory number 2, the lost civilization. This one is just as wild. Look at the map of carriers. The Basques, the Ainu of Japan, the Yanomami. These are some of the most unique, isolated populations on Earth. Their languages have no connection to any others. They are themselves genetic mysteries. What if they aren't randomly scattered? What if they are the last survivors? This theory pulls from Plato's story of Atlantis, an advanced prehistoric civilization on an island larger than Libya and Asia together, located beyond the Pillars of Hercules. A civilization that, according to Plato, was destroyed in a single day and night of earthquakes and floods 9,000 years before his time. That's 11,600 years ago. Long before the AI's 5000 BCE date for Elytra. What if the AI is wrong about the date? What if Elytra is older? What if it's the bloodline of the Atlantean survivors who fled the cataclysm in their ships and settled in the most remote corners of the globe? The spirit blood wouldn't be a mutation. It would be a relic, a piece of advanced ancient biology from a lost mother civilization. Wild theory. Number 3, directed panspermia. This is the one that has real scientists sweating. Forget ancient gods. Let's talk about real aliens. The theory is called panspermia, the idea that life on Earth didn't start here but was seeded from space, carried on meteorites and comets. But there's a more specific version, directed panspermia. This idea was championed by, of all people, Francis Crick, the man who discovered the structure of DNA. He and his colleague Leslie Orgel proposed that life on Earth might have been intentionally seeded by an advanced alien civilization. Sent here on purpose. So here's the theory. The Elytra glycoprotein, that invisibility cloak, isn't a human protein. It's the seed, an alien-designed molecule or even a microorganism that was sent to Earth billions of years ago. It latched onto our DNA and has been hiding there, dormant for eons. The epigenetic switch isn't random, it's an activation sequence. The AI in Paris, by finding the pattern, just stumbled on an alien program that's been running in our code for millennia, the shadow immunity. That's the point. It's designed to protect the seeded humans. The T-cell erosion? Maybe that's the program's life cycle. We aren't just a species, we're a project. This is what the scientists in Paris are grappling with. And it leads to the final, most terrifying theory of all. What happens now? In October of 2025, just 3 months after the AI's discovery, the International Society of Blood Transfusion held its emergency congress in Vienna. The mood was electric. The French team presented their findings, the data on Guada-1, the global map, the spirit blood of the Andaman divers, the shadow immunity, and the T-cell erosion. The room was stunned into silence. This wasn't just a new antigen, this was a fundamental rewrite of human biology. By the end of the Congress, it was official. The ISBT greenlit Elytra. It was the first major addition to our understanding of blood since the Rh factor in 1940. Every medical textbook on Earth was now technically obsolete. And that's when the real problem started, the what now problem. First, the practical, how do we keep these people safe? The 47 known carriers are just the start. How many thousands or tens of thousands more are out there? Every blood bank on Earth would need to rescreen its entire supply. But the tests are complex and expensive. You can't just use the simple ABO test. You need a genetic sequencer or a flow cytometer. The blood banks are scrambling. They are hoarding the few units of Elytra-positive blood in cryogenic freezers in Lyon, France. But it's not enough. For now, the only safe transfusion for an Elytra carrier is an autologous one, meaning they have to donate their own blood months in advance and store it. What if they can't? Then, the ethical nightmare. What happens when we find them all? Do we create a registry? An Elytra-positive database? What does that mean for their lives? Would they be barred from certain jobs? Would their insurance rates skyrocket? It's a whole new category for people to be divided by. But here's the catch. The other side of the debate is just as loud. The military and pharmaceutical companies are watching. They see the shadow immunity. They see the 87% neutralization of SARS-CoV-2. The race is on. Can we copy it? Labs at MIT and Stanford are already running simulations. Can we use CRISPR gene editing to mimic the Elytra coil? Could we create a vaccine that teaches the body to build this invisibility cloak for its cells? The models project a 40% resistance to Ebola. It could end pandemics. But what if we copy the curse too? What if we create a generation of super immune humans whose T cells just fade to dust? And this brings us to the final, wildest theory, the one that loops right back to the AI in Paris. Wild theory number 4, the glitch in the matrix. This theory says the AI didn't find a new blood type. It found a bug in the code. The simulation hypothesis, proposed by philosopher Nick Bostrom, is a serious academic idea. It suggests that if any civilization ever gets advanced enough to run ancestor simulations, basically a super realistic video game of their own past, they will likely run billions of them. If that's true, the odds are almost 100% that we are in one of those simulations. So what if the 47 people aren't people? What if they are glitches? What if the Elytra blood type is just a line of bad code? A rounding error in the physics of our reality that makes their biology act weird? It explains everything. The impossible immunities, the strange T-cell error, the bizarre global distribution. They aren't connected by ancestry, they are just random bugs popping up on the map. And the most chilling part? It took a machine to find them. The AI in Paris, a non-human intelligence, wasn't fooled by the simulation. It was just one computer reading another computer's code. It saw the bug and flagged it for its human operators. This is where we have to step back. People watching this are looking for a mystery, and this is the biggest one. So is this new blood type a blessing or a curse? Are we an alien experiment or just a computer program? Let us know what you think, and don't forget to like and subscribe.